Good afternoon. I'm Frank Szczepanski, CEO and co-founder of IVD Diagnostics. I'm here today to tell you about who we are, our revolutionary product, and why the market needs this technology now. We've raised $1.5 million to date and are currently seeking $5 million for a Series A round. Our revolutionary product, IVDX, is a system that utilizes our proprietary nanobioparticles which attach directly to tumor cells. They fluoresce, and we are able to read the circulating tumor cells through the patient's vein as they pass a peripheral point, a static point, on the back of their hand or arm. At the end of the scan, about 30 minutes, we've actually been able to monitor 1 liter, or about 25% of the blood in your body, without taking blood. Our nanobioparticles are non-toxic and created through the use of aptamers. Aptamers have a distinct advantage, in which case they're easy to modify, quite stable, have very high affinity, and easy for us to implement for multiple assays. At the end of that scan period, a report like this, either graphically or numerically, will actually pinpoint the number of CTCs that were scanned in the patient's blood. From a clinician's perspective, we have quicker turnaround time, saving a day or two in terms of time of report for either a biopsy or an imaging test. There's less risk, less bleeding, no hematoma, quicker and less costly test, and allows us to actually molecularly analyze the captured CTCs. In today's methodology, a patient would need to have one of these images done to be able to find a solid tumor. The problem with that is that solid tumors generally are not found until late stage 2. In fact, 75% of patients are diagnosed at too late of a stage. Our circulating tumor cell system actually can find cells that are down to the size of 2 to 20 microns in size, which these systems cannot. Some of the other disadvantages related to CAT scans and PET scans are their cost. It's generally about $5,000 to $8,000 in cost. Patients are also exposed to radiation. And the imaging size, as we had discussed previously, is much less sensitive for a molecular diagnosis. With needle biopsies, you have a similar problem. It's about $1,000 to $2,000 for a biopsy. Turnaround time is about the same, 1 to 2 days. It's often difficult to get a tissue sample, and it's not 100% guaranteed. If you'd like to see how our platform works, I would encourage you to go to our website, click on Technology page, and you'll actually see a video called How the Platform Works that you can click on. It'll give you a self-paced tutorial. Back in the spring of 2010, we actually conducted a live trial on 12 cancer patients with partners such as Franciscan Alliance and Indiana Hospital. It has about 30 hospitals in its hospital system. What we did is at the beginning of the trial, we actually, in the black columns, we actually measured what the patient's count was before their round of therapy began, and then 4 weeks later measured what had changed. And if you notice, there really isn't any average or threshold that's common across all these patients. Patients could have numbers that are small in the 20 CTC range per ml to one patient that was higher than 500. What's most important is the change. So if a patient had a count of 100 and it moved to 140, that was a 40% increase in their CTC count. It's the change that allows the physician to know whether or not the therapy is working. IVDX also works across multiple metastatic cancer types. We did a study with another medical center about 2 years ago, looking at 60 cancer patients and 11 different cancer types. These are the prevalent metastatic ones. And what we found is that virtually all of the major metastatic ones that are most prevalent, breast cancer, prostate, lung cancer, bladder, kidney, ovarian, and melanoma, can all be used with our test. Another important study we did was a linearity study together with a CLIA lab known as Alverno Clinical Laboratories. We actually found that we could measure down to a fraction of a cell all the way up to about 5,000 cells in 1 ml of blood. Give you a brief updating on our funding. We've actually raised $1.5 million to date. It came from private individuals, friends and family, and grants from the SBIR and matches from the state of Indiana. Now with the launch of our Series A, we're offering 2 million shares at $2.70 a share, which will net the company about $5 million. And the groups that have committed thus far include the St. Louis Archangels. We've also applied to the Indiana Elevate Ventures for matching angel funds from Indiana. We've also got pretty strong commitments from people on the Eastern Seaboard, including the Keiretsu Forum and the Archangels. One corporation who's willing to put in 6 figures of up to $1 million, and one corporate pharma who wants to follow on post the pilot trial. The way we would use our funds are predominantly in this way. About 80% of the funds will be used for both the pilot, which will cost us slightly above $1 million, and the pivotal of 2.4. What I'm asking for your help on is, let's get enough funding rounded up so we can begin the pilot in June of this year, be finished with that within 45 to 60 days, and in October we'll launch the pivotal. If we complete both of these trials, we can begin field deployment at the end of this year. We've looked at how many hospitals we need, minimally 4. We've got IRBs in place at 2. We're adding 4 additional hospitals. We have a CRO agreement in place, a general contractor who is committed to handling all of the data management, a supply chain company that will produce our proprietary reagent, as well as all the other items that are required for the trial. We have a biostatistical analysis company in place based out of Indianapolis. And the way the trial works is there'll be 100 cancer patients involved in the pilot and 200 cancer patients involved in the pivotal. I couldn't do this alone. We have a really great, outstanding team. We've been involved in a lot of activity, including both medical centers as well as business competition. For example, we won the Olin Cup for the Life Sciences business category at Washington University 2 years ago. We've teamed with notable scientists such as Dr. Vaclav Szczepanski, who's a Nobel Prize nominee. We recently won a Chinook Award from the Northwestern Indiana Society of Innovators, and the Regional Small Business Development Corp deemed us as the Revolutionary Technology Company of the Year. So we have a superior team comprised of scientists and managers, outside board of directors, medical doctors, and we're led by an accomplished CEO who thus far has closed $1 billion of business, and he's had 9 startups with 4 of them exiting with a valuation of $10 billion. When we take a look at the market today, it's about $44 billion when you look at the combination of both in vitro and in vivo diagnostics, with major players being some of the most notable pharmaceutical and technology companies, which also play a role in our strategy for potential licenses as well as exit strategy. And if you take a look at just the in vivo market, we can create a new market that we call CTC monitoring. If we take a look at the combination of both new patients, globally at 13 million plus about 80 million existing patients, we have a potential market of 93 million patients. If you take a look at just a small one-time scan per year, which is quite unlikely, you'd probably want to monitor at least 5 times a year. But if you looked at one time a year, it would be a $10 billion market. And by monitoring 4 times a year, we would generate about $30 billion in revenue. To get started, we focused on the Midwest. We have a number of early adopters, both medical hospitals as well as a CLIA lab called Alverno. Early demand shows the need for about 20 to 60,000 assays per year just from those early adopters, which would generate about 8 to 24 million in revenue. So why IV diagnostics? Well, we're focused on metastatic cancer, We're an in vivo platform that's extensible beyond oncology to other diseases such as inflammatory disease and infectious disease. We have the flexibility of using multiple small molecules based on aptamers, a broad field of use. We're totally dedicated to non-toxic diagnosis as well as treatment. And in effect, we're a solution that's called a new form of liquid biopsy. So I'd like to thank you for your attention, and I encourage the angels and the VCs in the audience to really take a closer look at investing in IV diagnostics. Thank you very much.